Okay, now let's get into a bit more depth regarding some of these processes. Write in your notes that transcription is the DNA-directed synthesis or building of RNA we call RNA synthesis. Now remember that proteins have many roles in the body, perhaps the most important of which is that they function as an enzyme. Well, write in your notes that the synthesis or building of the mRNA molecule in the nucleus is performed by the enzyme RNA polymerase, which pries the DNA strands apart and hooks together the RNA nucleotides. Okay, wait. So the building of a protein molecule is actually facilitated by an already formed protein working as an enzyme? Yep, that's correct. Can you tell me how we know polymerase is an enzyme? If you said because of the ASE ending, you'd be correct. Okay, now remember, this process follows the same base pairing rules as DNA, except that in RNA, uracil substitutes for thymine. So make sure you get that in your notes. So it shouldn't surprise you that there are several kinds of RNA. Let's get them and their functions in your notes. So reproduce this chart in your notes to refer to later on in this podcast. In case you haven't realized it by now, protein synthesis is a very complex process that uses many different molecules that all need to be constructed inside your cell. Now, where in the world does the cell get all these instructions? If you said DNA, you'd be correct. More specifically, the sequencing of nucleotides in each person's DNA. It's also worth mentioning again and writing in your notes that the actual construction of a protein occurs in the ribosome of the cell. So the real task is to get the instructions for making a protein and the raw materials for making that protein to the ribosome. And that is a completely complicated process that we're going to try and simplify. But here now is we're outlining the differences between RNA and DNA. So it says RNA is single-stranded, DNA is double-stranded. RNA is short, only one gene long, where DNA is very long and contains many genes. In fact, we'll do a DNA extraction from a strawberry this year, and you'll see how much DNA there is. RNA uses the sugar ribose instead of deoxyribose. And then, of course, RNA uses the base uracil instead of thymine in DNA. Now here are your different RNAs. So I want you to make sure you get this little chart in your notes to refer back to. And I'll let you write all the descriptions, and then as we move along, we'll describe each one more carefully. Okay, so let's try and break this process down so we can understand it a bit better. It's important to remember that it's both the instructions to build a protein and the amino acids necessary to build it that need to get to the ribosome before a protein can be manufactured or synthesized. We're first going to focus on getting the instructions from DNA in the nucleus to the ribosome. Write in your notes that transcription is the process by which DNA gets copied and an mRNA transcript gets made that then takes the copy out of the nucleus into the cytoplasm. Also get in your notes that because the transcription comes first, we call the first RNA that is made an RNA transcript. And there are 3 stages of this transcription process we need you to write down. And you see them here: initiation, elongation, and termination. Now, we're going to get to more specific explanations of it here in a second, but here's a general idea now of each stage. So don't write this down. We're just going to give you a kind of a preview. In initiation, RNA polymerase binds to the promoter. We'll talk about what a promoter is here in a second. The DNA strand unwinds. Remember, it's double-stranded. It needs to unwind. And the polymerase initiates RNA synthesis at the start point on the template strand, which you see right here. You also see now that they're labeled 3' and 5'. We'll talk about that here in a second as well. Then during elongation, polymerase moves downstream, unwinding the DNA and elongating the RNA transcript 5' to 3' direction. Write that down because it's going to be important. In the 5', right here you see the 5' to 3' direction. So you can see it, that RNA polymerase pointed that way. So it's going in the 5' to 3' direction. And it says here, in the wake of transcription, the DNA strands reform. All that means is, as this thing moves this way, the helix closes back up behind it. So it might be a good idea to write that down. In the wake of transcription, DNA strands reform a double helix. So you can see right here now that that has closed up. That's what they're talking about. Then eventually in termination, the RNA transcript is released. Here you see it being released, and the polymerase detaches from the DNA. So that's a general overview of the synthesis of an RNA transcript. Let's talk about them more specifically now. First, let's talk about initiation. Again, remember that RNA polymerase is the enzyme that will copy the instructions from DNA in the nucleus. Okay, first write that RNA polymerase binds to the promoter or start point, and that promoters signal the initiation of RNA synthesis. You see the promoter right here in this diagram. So here's your promoter. Now, also write now that the DNA sequence called the promoter is called a TATA box, T-A-T-A. T for thymine, A for adenine. So it's called a TATA box. So the other description of a promoter is the TATA box. Okay. Now, also get in your notes that next the DNA strands unwind and the polymerase initiates RNA synthesis at the start point on the template strand. Now, it's also important to notice that these DNA strands are numbered, and we talked about that. 3', that's how this is read, 3', and 5'. Now remember that we're going to copy DNA from the 5' to 3' direction. 5' to 3' in this direction. You see that now here in this diagram. 5' to 3' direction. Also notice that in the bottom picture, that DNA polymerase begins to copy the DNA strand after it unwinds. You see the new transcript being made right here. So here's the new transcript. Right here. Here's the new transcript. So as this polymerase moves this way, it's now leaving now the transcript behind, and it's now becoming longer and longer and longer as it reads now those sequences. Okay, now on to step 2, which is called elongation. And we're also going to include termination in this explanation, but elongation first. Write that in this step, RNA polymerase synthesizes or builds a single strand of RNA using the 3' side of unwound DNA as a template strand. We call that the anti-sense strand. S-E-N-S-E, anti-sense strand. So you can see now the direction of transcription, called downstream, Later on, we're going to talk about transcription that occurs upstream, but for now, we're talking about downstream from the 5' to 3' end. Now, also write that as RNA polymerase moves along DNA, it continues to untwist the double helix, exposing about 10 to 20 DNA bases at a time for pairing with RNA nucleotides. So RNA, remember this big, huge molecule just blown up is all it is, continues this direction, here's direction of transcription, and each time exposes 10 to 20 of these base pairings to be copied. Now you see uracil here that replaces thymine. So that's how we know this is an RNA transcript. Okay, finally on to termination. Write that specific sequences in the DNA signal termination of a transcription, and that when one of these is encountered by the polymerase, the RNA transcript is released from DNA and the double helix can unzip again. So just like it encountered a promoter, the TATA box, and it knew when to start copying, it encounters a termination sequence and knows when to stop and release the new transcript. Okay, so a quick overview of the transcription process. You see here where the RNA polymerase encounters the promoter or TATA box, signaling it to begin the transcription or copying process. So here's our promoter. Now it doesn't show the TATA, but you would expect a TATA type sequence here as a promoter. Once this process begins, RNA reads the code for each nucleotide on the 3' end of the unwound section of DNA, but we're moving in the 5' to 3' direction. It then begins to build the RNA transcript according to the sequence of each nucleotide contained on the DNA strand. And you see that happening over here in this little blow-up picture. It's important for you to write that codes for most amino acids are in what's called triplets, triplets, meaning it's a sequence of 3 nucleotides that codes for a specific amino acid on the DNA strand. Also get in your notes that this process is referred to as RNA synthesis because we're building an RNA molecule. Get in your notes also that for you that RNA polymerase knows to stop reading these sequences when it encounters what's called a terminator sequence, and you see that now down here. So just like the TATA box, the terminator sequence now would tell RNA polymerase where to stop. So, the end result is a piece of RNA called messenger RNA, or the RNA transcript, that now has all the information it needs to build a protein that's copied from DNA. So we've gone from DNA, and now we've copied messenger RNA, our first transcript. And it will take now the information from DNA out of the nucleus into the cytoplasm. If you don't already have that down in your notes, please write it now. So remember, ultimately we want the instructions that are in the DNA and the amino acids which are out in the cytoplasm to arrive at the ribosome so that we can have the instructions and the raw materials to build a protein. Okay, that's it for part 2 of protein synthesis. So, copy those notes, submit them to Moodle, pick up the study guide, and then answer those questions. And then I'll see you again for part 3 of protein synthesis in our next podcast. Bye.